So we left off briefly referencing the hormonal control of urine production, or the hormonal control of reabsorption. And you've seen this example a number of times. ADH, which is antidiuretic hormone, prevents diuresis. Antidiuretic hormone prevents diuresis. And the typical negative feedback loop that you often see referenced is shown here. So if osmolality or osmolarity goes up, and those are two different things, I actually bring that up today. If osmolarity goes up, fluids are more concentrated. We need to dilute those fluids so we reabsorb more water. And this is a really nice example to understand the basic negative feedback. But in practice, it's never as clean cut as this. It's not so easy as saying, oh, my body fluid osmolarity is up. I better release ADH. Often, it's up and volume is up, or volume is down, and these levels fluctuate. And so release of hormones like this is not on or off. It's not present or it's not. It's always changing slightly. So keep that in mind as we talk about another layer of hormonal control. On the last slide, we have this single stimulus where osmolarity is increased, but a more functional, applied example might be if blood volume is lower. Blood volume might be lower if you're dehydrated. Blood volume might be lower if you hemorrhage and lose a lot of blood. If blood volume is low, this is a really important disturbance to correct. Blood volume is too low, you can't have blood flow, and you pass out, you faint. You can't escape from danger if you're being chased by a tiger, who knows. So you want to correct low blood volume. And we saw yesterday's class, or even Tuesday's, I can't remember, there's so much information, how the kidneys release renin in response to a change in pressure at the juxtaglomerular apparatus. And you've already seen renin referenced a few times. I brought this up at the start of this section. And this was reprised from the blood pressure section way at the start of the semester. The kidneys release renin in response to low pressure, indicating low volume, and then a whole series of cascades happen. The cascade is shown here. Angiotensin 1 converted to angiotensin 2 by angiotensin converting enzyme. Aldosterone is produced. And then things happen to help restore this bad situation, low blood volume. One of those things that happens is angiotensin 2 constricts the afferent arterioles to limit filtration. Well, that makes sense, right? If body fluid is low, if blood volume is low, I don't want to get rid of any extra fluid or solutes. I don't want to filter out anything that I'm trying to conserve. So yeah, I want to limit GFR, which is what angiotensin 2 does as it vasoconstricts. Angiotensin 2 also happens to affect ADH release. Angiotensin 2 will prompt ADH production. So that negative feedback loop that you saw on the last slide also happens. We try to limit the filtration of stuff we're trying to hold on to, and because blood volume being low is bad, well, I'm also going to turn on ADH. That's going to bring more water back in, allow me to reabsorb more water, and hopefully restore this reduced blood volume scenario. So you already know the actions of ADH in the distal convoluted tubule. It opens up pores and brings water back in. That, in conjunction or in addition to actions of aldosterone, help to conserve the important fluid and solutes that would otherwise be excreted. Now, we haven't spent a lot of time talking about aldosterone here. You've heard about aldosterone. Like ADH, reabsorbing water, aldosterone helps to reabsorb sodium. Very similar actions in that sense. And by reabsorbing sodium, we help to accentuate the pull of water back into the body. Overall, conserve, conserve, conserve is the message of these hormones. Low blood volume means don't get rid of any fluid, don't filter out any solutes, and conserve anything that has been filtered out. I'm at a critical juncture. Blood volume's low, I'm about to pass out. So I like that example because it gives us a nice, it illustrates the two-pronged approach to a more complex problem. We're not just reabsorbing sodium, we're not just reabsorbing water. By doing both, we fix the low blood volume issue.
Hormonal control of water retention also occurs in the nephron. Extracellular osmotic pressure is regulated by the presence or absence of antidiuretic hormone, ADH. When the blood plasma becomes too concentrated, release of ADH signals the formation of water channels in the distal convoluted tubule and collecting duct, allowing the kidneys to reabsorb additional water back into the bloodstream. When ADH is not present, excess water is excreted from the body as dilute urine. Blood pressure and volume are also affected by the kidneys. If receptors in the afferent arteriole sense that blood pressure is low, the juxtaglomerular apparatus releases an enzymatic hormone called renin. The lungs respond to the release of renin by producing an activating enzyme, which results in formation of angiotensin II in the bloodstream. Angiotensin II causes blood vessels to constrict, increasing blood pressure, and stimulates the adrenal glands to release the hormone aldosterone, which also causes an increase in plasma volume and consequent blood pressure. Aldosterone causes greater plasma volume because it increases active transport of sodium and chloride ions into the interstitial fluid. Combined with the production of ADH, the additional reabsorption of sodium and chloride ions causes more water to be drawn into the bloodstream by osmosis, resulting in increase in blood volume.
All right, you've been ADH'd to death, I'm sure. So this is low blood volume or low body fluids. In lab 8, we saw something different with large ingestion of fluids. You saw a surplus of body water. And incidentally, if you're a beer fan, Unfiltered, best brewery in Halifax. If you happen to be there ever on a weekend, stop in. Absolutely the best beer. So we see this oversupply of fluid. Maybe it's beer, maybe it's cranberry juice, maybe it's tomato juice. Large fluid ingested. And I like to think of a systematic approach to thinking about the effects of each of these beverages would be to consider the control first. What would happen if just fluid was added, just water was added? In a pure sense, you're adding 700 mils, 800 mils, whatever the volume is, of plain water to the fluid compartments in the body. That would serve to increase the volume and dilute those compartments. It would sense it in the blood, but it would also do this to the interstitial fluid and to the cells in some respect. But overall, the main effect would be to reduce osmolarity and increase volume, blood volume or blood pressure. So the response should be the opposite of what we just talked about, right? We just outlined low body fluids, and in this situation we're faced with probably higher body fluids than we would be expecting, or that we would like. And so we get rid of it. We measured how much we got rid of it during 2 hours of lab. The response is limit reabsorption, get rid of the extra fluid. But how do we achieve that? How do we limit reabsorption? How do we get rid of the extra fluid? It's not a crisis, but the signal is to produce more dilute urine. And that's what we're going to talk about more in depth today. So things like ADH aren't released. ADH is not prompted when osmolarity drops, when body fluid goes up. And in fact, ADH release, if you're measuring it in the blood, can drop to near zero. And that doesn't mean push more water out. It simply means I don't want any part in reabsorbing water in the distal convoluted tubule. Renin's not going to be activated, so no aldosterone is released, therefore no sodium is reabsorbed either. Anything that is in the distal convoluted tubule should stay there and eventually be excreted. I guess in theory what would happen is we would shut off all reabsorption after the nephron loop. And so if you go back and look at the percentage of reabsorption that I tried to outline through each section yesterday, when we leave the nephron loop, we've reabsorbed 80-ish% of what was filtered out. And in this situation, that remaining 20 would ideally be excreted. It's not going to be 20. Some does get reabsorbed, but we're trying to push the envelope and excrete as much of that as possible because we have a surplus. So understanding this condition, what normal fluid would do, what plain water would do, the other beverages modify this condition. It's the same, it should be a similar volume, different people, different volumes, but the stimulus should be similar. You're taking in a large volume of fluid, but that fluid now has sodium in it, or that fluid is now acidic, or that fluid is mixing with the Tums that you force down your throat and is now basic. And so if we consider that control condition, observing how each of these individual beverages changes the net outcome might give you some insight into how each of these will individually impact reabsorption. Okay, I'll leave that for you to decipher in your lab exercises. I'm answering along with you. So let's talk about how urine's produced. In this scenario, urine's produced. We know that. We can observe it, but we don't understand the mechanism yet. So urine production always happens. Unfortunate side effect of life. You can hold it for as long as you want to, but you're never going to stop urinating. Urine is always produced because fluid intake is highly variable. There wasn't anything on here to miss. You all right? Fluid intake, highly variable. I don't think we saw anyone. Ooh, you can't really see this. We saw no one down in this end, the really dark, cloudy urine that's labeled as consult a doctor, luckily. Fluid intake, highly variable. We want to maintain normal body volume, normal body osmolarity. Because fluid intake is always variable, we're always changing body fluid and body osmolarity. Some of the fluid gets lost through sweat, some of it through respiratory water loss. There's a lot of different ways that you can lose body fluid, but one of the major ones at rest is through urine. And urine production is calibrated to maintain body fluids at a specific level and a specific concentration. But it's a pretty versatile system. If you only sat here and typed, and if you only drank water, you'd only need to produce one kind of urine. But urine can both be concentrated, it can be dilute, it can be in large volumes, it can be in small volumes. And it can change throughout the course of the day. How is it that one system can do all of that? We'll color around the edges first. We already introduced this case. If you have a large fluid intake and you increase body fluid volume, the response should be let out a large amount of dilute urine. The control condition in lab A. Large fluid or water intake increases body fluid volume should result in a similarly large output of urine. We don't have any room to store that extra water. We need to keep body compartments at an appropriate volume and concentration. On the other end of the spectrum, small fluid intake means body fluid decreases. We don't want that situation. Long term, we want to conserve fluid in that situation, so we will release small amounts of concentrated urine. Those are the two ends of the spectrum. How is it that one system, that the nephrons, can do both dilute urine production and concentrated urine production? I should mention, you've probably picked up on this already because we've had 2 slides with the Turning Point logo on them. But this is a little hint to help you, saying, look to the questions, you can answer one of them with the information on the slide. You figured that out. The reason why the nephrons, the kidneys, can make urine of different compositions is due to the countercurrent multiplier. This is why, or how, the system works. We don't understand what it is yet, but it's because of this phenomenon that we can make both dilute and concentrated urine. So countercurrent simply describes flow in opposite directions. And the flow that I'm talking about is that in the nephron loop. You've seen that long loop extending into the medulla. There's a hairpin turn, and it ascends right back up, close to, next to the descending limb, but flows in opposite directions. That's the countercurrent aspect. And you know, because we mentioned it, that the ascending limb specifically will pump out solutes. Water stays in the ascending limb because it is impermeable to water, and therefore the surrounding environment gets more concentrated. Solutes go out, fluid doesn't. More solutes out means more higher concentration. And it's really here that the countercurrent multiplier starts. The gradient is created by the ascending limb, and it is imposed onto the tubular fluid that's coming in to the descending limb. And it's because they're in such close proximity that the phenomenon of increasing osmolarity can have an effect on the tubular fluid. So we're going to look in detail on how the gradient is created and how it's imposed. Let's start at the beginning. Fluid coming in to the nephron loop has a very similar concentration and constituency to blood. The way that we measure total concentration, you know, is osmolarity, and blood is around 300 milliosmoles per liter. The reason that I keep switching between osmolarity and osmolality is one simple distinction in how these are each measured. Osmolarity is total solutes per liter. Osmolality is total solutes per gram or per kilogram. And you can measure volume either by titrating it in a beaker, or you can weigh it on a scale. In all of my research, I weigh it on a scale, so I default to osmolality. If you hear me using one interchangeably, it means the same thing, total concentration. So, blood coming in around 300 milliosmoles per liter. The filtrate that enters the proximal convoluted tubule, again, about 300 milliosmoles per liter. And that continues until we enter the nephron loop. The cortex is around 300 milliosmoles per liter. The tubular fluid is about 300 milliosmolar. And now we enter a situation where that changes. The osmolarity changes because the concentration gradient in the medulla has been established previously. This is now modified as fluid and solutes move between these two compartments. And the gradient on this figure isn't mistakenly darker near the bottom. It's because concentration tends to be higher near the bottom. So, at the start, there's no reason for there to be any difference between these compartments. There's free exchange, and the total concentration is similar. Down here, there is a very distinct gradient, and we get fluid and solute movement accordingly. In general, tubular fluid gets more concentrated the deeper it goes. As it ascends out of the medulla, it starts to get more dilute, and then it gets even more dilute in the distal convoluted tubule. So osmolarity increases as you go down, decreases as you go up, and decreases further as you leave the medulla. Why does it increase when you go down? And why does it decrease as you go up? Why is there a variance? Why does the concentration change? The reason that it changes is thanks to the impermeability in the ascending limb. We know well there are transporters for sodium, potassium chloride, and those are leaving the tubular fluid. There's low water permeability, so the net effect is as fluid rises in the ascending limb, its concentration goes down. Ions are being pumped out, fluid can't leave, tubular fluid is more dilute. Okay. What does that do to the immediate surroundings? Well, the immediate surroundings are what accept those ions. We now have a higher concentration of ions immediately surrounding the ascending limb. And this keeps going. And more ions are pumped out. No water can join them. And so we get this large, high concentration, high osmolarity environment. A very concentrated environment. If this were all there was, it would be pretty straightforward. But we don't like this situation. The body doesn't want to have really high concentration compartments. It wants everything to be even. Water wants to move so that it dilutes this environment. It can't move out of the ascending limb, but it can move out of the descending limb. because the environment is more concentrated, it promotes water leaving the descending limb. What effect is that going to have? Water leaving the descending limb would serve to concentrate the tubular fluid. So this is in addition to the ions and fluid movement that we discussed yesterday that occur in the descending limb. Now we have this environment where there is a higher concentration in the medulla that promotes the reabsorption of water. Water wants to move to quench the really concentrated interstitial fluid in the medulla. So we have this large gradient initially. As soon as fluid comes into the medulla, water rushes out. That starts to increase the concentration of the tubular fluid. And in doing so, if I were to ask you, okay, gradient for water to move out of the tubular fluid, is it a large gradient here at the start of the nephron loop? High concentration around, large gradient for water to move out. Yes. Is it larger here than it is further down in the nephron loop? The draw for water to leave, for water to go into the interstitial fluid. Yes, it's much higher initially because the concentration going up means there's less of a gradient for water to follow out of the tubular fluid. The lower stimulus to reabsorb water is evident as you go down the nephron loop. So how does this create that vertical gradient? Filtrate enters the nephron loop. All of a sudden, I'm in this really salty, concentrated environment. Water wants to leave, and it does because there's a very high gradient. So I get lots of water movement out up here. Lots of water movement out means I'm slightly more concentrated. I continue going through the descending limb. Water continues to move out, but there's less of a gradient. I concentrate a little bit more. Water moves out again. Water continues to move out. I progressively get more and more concentrated as I reach the tip of the nephron loop. There's more dilute fluid up here, more concentrated fluid at the bottom, and in such a way, we have this gradient from top to bottom with low concentration to high concentration at the tip of the nephron loop. And we end up, if we're comparing to the 300 milliosmoles that are incoming, there might be 350 just across the border, 550, 750, 900 in the tip. And then as we pump solutes out in the ascending limb, gets more dilute, and we eventually reach a very similar concentration of the incoming fluid. But note, this is more dilute than the incoming fluid was. We pump out more solutes than we need to, and we dilute this fluid a lot. So the impermeability in the ascending limb is what makes the surrounding fluid concentrated. There's a quick rush out of water here, and that trickles to a stop as you reach the tip of the nephron loop. It gets slower and slower, making this vertical gradient. Lower concentration up top, higher concentration on the bottom. And so if we were just to form urine, let's make urine. The collecting duct right now would expel anything that it was given. This is a situation where we have high body water, we don't need to reabsorb any, ADH isn't influencing anything, there's no aldosterone. There's low water permeability in this region because we don't have ADH, so most of it continues to filter through. Urine is dilute because we still continue to reabsorb some solutes. Solutes being, solutes being sodium, chloride, and some urea, which we'll come back to a little bit later. As we continue to reabsorb solutes and not water, notice the concentration drops. We end up producing large volumes of very dilute urine. Because we have this concentration gradient that made for a low osmolarity tubular fluid, and then we continue to remove solutes, we expel a large volume of dilute urine at the end of this process. But how then does this system alter or change when we don't want to produce dilute urine? How can the same series of events produce concentrated urine? Concentrated urine is mostly produced in those really long nephrons, juxtamedullary nephrons. And for all intents and purposes, I'm using the same schematic. There's a couple key differences. One of them is not shown here. The loop is longer. This is the same picture, so you're not seeing a longer loop. But you can imagine, with a longer loop, there's a greater concentration gradient, and so fluid in the tip here is 1,200 milliosmoles, not 900 like in the shorter nephron loops. So that's one important difference in making concentrated urine. Otherwise, everything else is set up the same. Incoming fluid has the same concentration. We see a progressive increase in that concentration in the descending limb. It spikes a little bit higher in the tip, but it also decreases as we return to the distal convoluted tubule. Same setup. So how is it that we produce concentrated urine and not diluted urine? The answer lies with ADH. All of a sudden, these pores open up. ADH promotes the reabsorption of water through these aquaporin pores. Water moves through. As water starts to move through, it concentrates the fluid in the collecting duct, and instead of dilute urine, where water stays in the collecting duct, we end up with concentrated urine. The large majority of it is reabsorbed. One important piece to connect before we end is, why does water move? I can open up an aquaporin. I can make a channel for water to move out of the collecting duct. Why would it want to move? Anyone have any guesses? Why would it want to leave? Especially in this region as we produce really concentrated urine. There needs to be, sorry? You want to what, sir? You need to keep the fluid in the blood, but what makes it go back into the blood? What is the draw for fluid to leave? I open up these channels. It's the concentration gradient. If this vertical gradient didn't exist, you wouldn't get nearly as much movement of water out of these pores. Because water wants to move, and it wants to quench the gradient in the medulla, we get large reabsorption of water and the ability to create concentrated urine. Without that gradient, we wouldn't have the ability to make as concentrated urine. Maybe this will help recap and is more clear.
To explain the physiology of the countercurrent multiplier, we will begin with the ascending limb of the nephron loop. It is important to note that the entire ascending limb is impermeable to water, but actively transports sodium, potassium, and chloride ions out of the tubule and into the interstitial space. As active transport of ions out of the ascending limb continues, the interstitial fluid surrounding the tubule becomes more concentrated. As a result, a vertical osmotic gradient is established in the interstitial fluid due to the high concentration of ions deep in the medulla, and gradually decreases toward the cortex to levels consistent with fluids in the rest of the body. The osmotic gradient in the interstitial fluid is measured in units of osmolarity, with larger numbers representing higher concentrations of solutes. On the other hand, the descending limb of the nephron loop is freely permeable to water, but impermeable to most other substances. As filtrate passes through the descending limb, water is drawn into the interstitial space via the osmotic gradient developed by the ascending limb. By the time the tubular fluid reaches the bottom of the nephron loop, housed deep within the medulla, it is at its maximum concentration, and the interstitial fluid is at maximum osmotic pressure of about 1200 milliosmoles. Recall that the ascending limb of the nephron loop is impermeable to water, but actively transports sodium, potassium, and chloride ions into the interstitial fluid. As a result, the tubular fluid becomes less and less concentrated as the filtrate descends back toward the cortex. The filtrate becomes even less concentrated than plasma as it reaches the distal end of the nephron loop. The net difference in tubular fluid concentration between the descending and ascending sides of the countercurrent mechanism results in a 200 milliosmol horizontal gradient. Overall, the filtrate becomes progressively more concentrated as it passes down the descending portion of the loop, and progressively less concentrated as it moves up the ascending loop. The countercurrent multiplier is responsible for creating and maintaining a vertical osmotic gradient within the kidney tissue. Its purpose is to provide a mechanism by which the reabsorption of additional water can be implemented.
Rapid fire. The tubular fluid changes concentration as it moves through the nephron loop. That summarized everything that we talked about. That is the countercurrent multiplier.
Explain the physics.
Nope, not again. Briefly, last 5 minutes, I want to address countercurrent exchange, which is a separate idea from countercurrent multiplication. This one is far less involved, and it has to do with blood. I'll pay lip service to this, and then we can call it for the day. So, there is a blood supply, obviously. Cells in the nephron need oxygen and nutrients, and blood has to be there to pick up anything that's reabsorbed. In those really long nephrons, the juxtamedullary nephrons, the capillary bed, specifically the vasa recta, make long loops a lot like the nephron loops. And the blood that travels deep into the medulla is subject to the gradient, just like the tubular fluid is subject to the gradient. Blood is subject to the gradient as well. We don't want blood to affect the gradient. Blood is completely subject to the gradient. It's modified by the gradient. The constituents will change as it dives into the medulla, so that the gradient is unmodified. The blood here wants to deliver nutrients, pick up substances that are reabsorbed. It doesn't want to undo all the work the kidneys have done in creating that vertical gradient. And so blood is malleable. The concentration, the volume will change readily as it dives into the medulla and then resurfaces. The next slide that I'm going to show you looks complicated. I'm delaying because I can hear a lot of clacking still. It looks complicated, but don't be scared. We've seen all of this already. There's a lot of words on it, and numbers, and arrows. But if you look, the left-hand side has all been explained. The left-hand side just tells you about the countercurrent multiplier. We've seen numbers like these before. We know that the tubular fluid gets more concentrated near the bottom and then less concentrated as it goes up. What we're highlighting on this slide is how this situation that we've already explored on the left compares to the blood on the right. And I just want you to notice, as blood enters, it is freely changed. Blood enters a concentrated space. Water moves out to equalize that concentration. And solutes move in that were reabsorbed. This is where they're supposed to go. As the concentration goes up, that's magnified, and it freely changes as it dives deep into the medulla. On its way out, the exact opposite happens. The surrounding concentration lessens. Water runs back in. Some solutes are transported out. But overall, the net effect is that blood is relatively unchanged, as is the gradient, which is fantastic because maintaining that gradient allows us to continue reabsorbing water and making concentrated urine if we need to. The blood constituents freely change as a result of the gradient. They don't impact the gradient that's established by the countercurrent multiplier. This phenomenon is called countercurrent exchange. Free movement of water and solutes in and out of the blood. Good? And that's all the questions. Have a wonderful weekend.